So good day again, everyone. So for this particular lesson, we will be talking about cell structure and taxonomy. Particularly, we will try to differentiate between the eukaryotic and prokaryotic cell. And of course, we will be summarizing the structural differences between these two kind of cells. And then we will also be tackling how these organisms and their cells would divide, the taxonomy, the evolution and the tree of life, and determining relatedness among organisms. Okay? So, as we all know, cell is considered to be as the basic unit of life. Okay? So it is considered to be as the fundamental unit of any living organisms. Okay? Because it exhibits the basic characteristics of life. Okay? So in fact, some microorganisms are said to be unicellular. When you say unicellular, when you say unicellular, we are actually referring to a single cell organisms. Now, for us, since we are not unicellulars, we are not microscopic, we're humans. So as we are considered to be as multicellular because we are made up of different cells. Now, as regards to the categories, there are two main types of cells, which I have already introduced way back in the introduction. So we have the eukaryotic and prokaryotic. Again, the term "eu" means two, while the term "karyo" refers to nucleus. So when you say eukaryotic, this one is provided with true nucleus. Okay? And what makes it true nucleus? Because of the presence of a nuclear membrane. So because of the presence of the nuclear membrane, then that is considered to be as a true nucleus. Now the term pro means before, which primarily means primitive. Okay? Therefore, when you say prokaryotic, this one refers to primitive nucleus. Okay, so some microbes are said to be prokaryotic, such as bacteria and archaeobacteria. These are examples of prokaryotic. Okay, some microbes are said to be eukaryotic, such as algae, protozoans, and fungi. Okay. However, some microorganisms, some microorganisms are not composed of cells. So meaning to say, when they are not composed of cells, it means that they are acellular. So it means that they are acellular. And since they do not have the basic unit of life, therefore we consider them as a non-biological entity. So they are considered to be as a non-biological entity. So an example of such would be the viruses, prions, and viroids. Okay. Now take note that even if they are non-biological entity, they are still considered to be as infectious. So they can still cause infections. As a matter of fact, the causative agent of COVID-19, which is SARS-CoV-2, is an example of a virus. So again, microbes are divided into acellular. So this includes the viroids, the prions, and the viruses, and the cellular, which can be classified as prokaryotes and eukaryotes. Okay. So I have already discussed this one to you. Eukaryotic contains true nucleus, whereas prokaryotic cells do not have true nucleus. And what makes it a true nucleus? because of this structure. The presence of, if this is the cell, then this is a nuclear membrane. Then inside the cell, inside the nucleus are the chromosomes. And the gel-like fluid found inside the nucleus, we call it the nucleoplasm. Okay? So this makes the cell a true nucleus. Now, another important characteristic of eukaryotic cells is that it composed of complex system of membrane and membrane-bound organelles. So sometimes this one is otherwise known as the membranous organelles. Okay. Prokaryotic cells, membranous organelles. Now, both eukaryotic cells and prokaryotic cells possess cell membrane. Cell membranes are very important, and the most important function of the cell membrane is for selective permeability. Okay, when you say selective permeability, it will only allow certain substances to pass through them. So, meaning to say, not all substances will be allowed to pass through a certain cell. And the reason why it is capable of the so-called selective permeability is because, chemically speaking, cell membrane is made up of lipid bilayer. Meaning to say, there are two layers of lipids that allows them to be semi-permeable. So the other term for this one is semi-permeable. So selected permeability means semi-permeable. Okay, so again, this is the transmission electron micrograph view of the cell. So as you can see, you'll be able to study the ultrastructure of the cell. Okay, so this is the TEM view. So what would be the basic structure of the cell? So the presence of nucleus. So this is the nucleus. And since it is made up of nuclear membrane, so the nuclear membrane is present. And then inside the nuclear membrane is the nucleoplasm. And different chromosomes are embedded in the cytoplasms. So this makes the cell eukaryotic. And aside from that, you'll be able to find different membranous organelles. And to name a few, we have here the mitochondria, we have here the lysosome, we have here the cytosol, the Golgi apparatus. Okay, so these are the different membranous organelles embedded within the cell. So let's talk about nucleus. So the nucleus is considered to be as the command center of the cell. And it's a command center because it directs, okay, the division, the cell division. Okay? And it also controls important mechanisms such as protein synthesis. Okay. So as what I've told you, it has three components, namely the nucleoplasm, the chromosomes, and the nuclear membrane. So where can we find the chromosomes? So chromosomes are embedded within the nucleoplasm. And why chromosomes are important? Chromosomes are important for our heredity because eukaryotic chromosomes contains linear DNA, molecules, and proteins. And as we all know, DNA, deoxyribonucleic acid, is considered to be as the blueprint of life. So the reason why you're short, the reason why you're tall, the reason why I'm bald, the reason why you have a kinky hair, is because it's all imprinted in our DNA. Primarily because the genes are located along the chromosomes. And an organism's complete collection of genes is referred to as the genotype or the genome. Okay? So each gene contains the information to produce one or more gene products in the form of proteins. So that's the reason why I told you a while ago, the nucleus is considered to be as the command center because it's responsible for cell division and for our protein synthesis. Okay. So although most genes code for proteins, okay, some code for two RNAs. So we have the ribonucleic acid, the ribosomal ribonucleic acid or the rRNA, and transfer ribonucleic acid or the RNA. In fact, another type of RNA is the mRNA or the messenger RNA. The number and composition of chromosomes and the number of genes on each chromosomes are characteristics of the particular species of organisms. So, for example, as humans, we have 46 chromosomes. Okay? We have 23 pairs. 22 are considered to be as the somatic chromosomes, while we have one pair of gametes. Okay. So it has been estimated that the human genome consists between 20,000 and 25,000 genes, which have already been completed. So our chromosomes have already been mapped through the human genome project. So the purpose of the human genome project is to map all the genes inside our body. Okay. So let's talk about structure of the eukaryotic cells. Okay, so we have, of course, the cytoplasm. Okay, so we have the cytoplasm. Okay, so the cytoplasm is a semi-fluid gelatinous nutrient matrix. So where can we find the cytoplasm? The cytoplasm is found, okay, inside the nuclear or inside the cell membrane. So it is found inside the cell membrane. Okay. So I think my audio was not audible a while ago. So I will go back. Okay. So again, some genes will code for RNA. Okay. So these are the ribosomal nucleic acid and the transfer nucleic acid. Okay. So the ribosomal nucleic acid is important, particularly for protein synthesis. And then the tRNA is important to transfer codons, which are important, okay, for the production of amino acids. Because each amino acid is made up of 3 codons. And the mRNA is important because mRNA will carry the message from DNA to the ribosomes for protein synthesis. Okay, so as what I told you, the number and composition of chromosomes and the number of genes on each chromosomes are characteristic of the particular species of organism. So it depends, no? But in cases of us humans, we have 46 chromosomes, okay? So we have 46 chromosomes, and these 46 chromosomes are made up of 23 pairs. So 22 pairs are actually called somatic chromosomes, while one pairs are actually made up of gametes. So it has been estimated that the human genome consists of between 20,000 and 25,000 genes, which have already been mapped so through the project known as the Human Genome Project. Okay, so let's talk about the different eukaryotic cell structures. So first we have here is the cytoplasm. So the cytoplasm is found inside the cell membrane. So physically speaking, it is made up of a semi-fluid gelatinous nutrient matrix. So if you will be examining the cytoplasm, you'll find out that it is made up of different storage granules and a variety of organelles. And each organelle would have a specific function. So the cytoplasm is the site where most metabolic activities of the cell is actually occurring. So that's why you can find here different kinds of organelles. Okay. Now, let's talk about in detail these organelles. Now, when it comes to endoplasmic reticulum, there are actually two types of endoplasmic reticulum. So we have the smooth and the other one is the rough endoplasmic reticulum. Now, physically, morphologically speaking, endoplasmic reticulum is a highly convoluted system. So it's like meshwork, okay, arranged to form a transport network in the cytoplasm. Okay? So how do we differentiate the rough ER from the smooth ER? The key word here is the presence of ribosomes. Okay? So rough endoplasmic reticulum has ribosomes attached to it, whereas the smooth endoplasmic reticulum do not have ribosomes attached to it. So the ribosomes are actually consisting of ribosomal RNA and protein. And this is considered to be as the site of protein synthesis. So the ribosome is the site of protein synthesis. Okay. Now we also have here the Golgi complex, or sometimes you will see the term Golgi apparatus or Golgi body, and they're all the same. So it's also a network, and it is directly connected, or it can actually communicate with the endoplasmic reticulum. So it completes the transformation of newly synthesized protein and package them for storage or export. Yeah. So if endoplasmic reticulum is the factory of protein, Golgi complex is like the packaging center. Okay, ready for the materials to be delivered, okay, to the sites where they are needed the most. Okay, so Golgi complex, okay, it's like the packaging center. So aside from that, we also have the lysosomes and peroxisomes. So the lysosomes and these peroxisomes have actually originated from the Golgi complex. So lysosomes contains hydrolytic enzymes and other digestive enzymes. And the purpose of these enzymes is to degranulate foreign materials such as the phagocytized bacteria. Okay, so they can degranulate foreign materials such as phagocytized bacteria. Okay, peroxisomes are another example of membrane-bound vesicles where hydrogen peroxide is generated and broken down. Okay, so usually peroxisomes will break down hydrogen peroxide into water and oxygen. Okay, so this is where hydrogen peroxide is broken down. Why? Because hydrogen peroxide is actually a super oxide radicals. And these are actually toxic for the cell. Since it is toxic for the cell, it is very important that we break it down. So peroxisome will break down superoxide radicals such as hydrogen peroxide because that is considered to be as toxic for the cell. Okay, so moving forward, we also have the mitochondria. Okay, it's considered to be as the power plants or the powerhouse of the cells or the energy factory. Okay, so why is it considered as an energy factory? Because this is where the ATP is being generated within the mitochondria via the cellular respiration. So if you will be looking at the structure of the mitochondria, you'll be able to see the inner folding within it known as the cristae. Okay, and do you know that mitochondria is the only organelle that it has own mitochondrial DNA? Mitochondrial DNA. And do you know there's another trivia that mitochondrial DNA is maternal by nature. So you actually get it from your mom. And that's why there's some sort of connection between a child and the mother because, you actually get your own mitochondrial DNA. Okay, so it's maternal by nature. So the number of mitochondria depends on the activities of the cells. So there are some cells, such as our muscle cells, that would require more mitochondria as compared to other kind of cells. Okay, plastids are membrane-bound structures containing photosynthetic pigments. So these are found in plants. Okay, so they are the sites of photosynthesis. So chloroplasts are types of plastids, and it's the one that contains chlorophyll, a pigment that is necessary for photosynthesis. Okay, and then cytoskeleton is another structure found in the eukaryotic cell, and it is a system of fibers throughout the cytoplasms. Okay, so it's like it serves as an important framework for the cell. Okay, and there are actually three types of fibers. Okay, so for example, we have the microtubules and microfilaments, and then we have the intermediate filaments. So let's say, for example, microtubules are important for mitosis or cell division. Okay, so let's talk about cell wall. Some eukaryotic cells contain cell wall. Okay, so for example, fungi has cell wall, and the name of the cell wall is known as the chitin. Okay, so it's an external structure to provide shape, protection, and rigidity. Okay, the cell wall is much simpler in prokaryotic cells. So, say chitin is found in cell wall of fungi. Cellulose is found in cell wall of algae and plants. Okay, so these are examples of eukaryotic cells with cell wall. Okay, so take note that cell walls for the eukaryotic cells, these are the eukaryotic cells. Bacteria are examples of prokaryotic cells. Okay, so eukaryotic cells have cell wall. eukaryotic cells have cell wall. Okay. Bacteria are prokaryotic and they also have cell wall. However, as humans, as part of animal kingdom, we do not have cell wall. Protozoans do not have cell wall, and mycoplasma is an example of bacteria without cell wall. So mycoplasma is an example of bacteria without cell wall. Okay? So let's talk about other eukaryotic cell structures that we can find. So we have here the flagella and cilia. Okay. So the flagella and cilia may also contain microtubules. So some eukaryotic cells, such as spermatozoa and certain protozoa, can possess long, thin, whip-like organelles of locomotion. So we call it the flagella. Okay. So they're important for locomotion or for motility. Okay. So flagellated cells may possess one or more flagella. So it could be a single flagella or it could be several flagella. Okay. Some cells moves by means of cilia, okay, which are shorter, thinner, and more numerous than flagella. So if flagella is longer, cilia is shorter, but they are more numerous. So they are shorter, but they are actually more numerous. So that's an example of a cilia. So cilia can be found in some species of protozoan. So an example of protozoan with cilia is called Balantidium coli. So Balantidium coli is the largest intestinal protozoan that can parasitize humans. However, some cells of our body are ciliated. So some cells of our body are ciliated, such as this kind of cell. So it is an example of ciliated cells in our body. So an example of that is the pseudostratified ciliated columnar there's a delay in yan, pseudostratified ciliated columnar epithelium. The pseudostratified ciliated columnar epithelium is actually found in the linings of our trachea. It's found in the linings of our trachea, and the function of that cilia is that it can propel away mucus. Okay, it can propel away mucus, and as it moves away the mucus, we are also removing bacteria from our respiratory tract. So it's actually part of our body defense. Okay, so that's the pseudostratified ciliated columnar epithelium found the linings of the trachea. Okay, so here's an example of a cilia, a cross-section of the cilia. Okay, so the cross-section of the cilia has a 9+2 arrangement. A 9+2 arrangement of the microtubules. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 plus another 2 microtubules here in the middle. Okay, so 9+2 arrangement of microtubules. Okay, so let's talk about the prokaryotic cell structure. So the prokaryotic cells are actually 10 times much smaller than eukaryotic cells. So prokaryotic cells are simple compared to eukaryotic cells. So they simply divide by means of binary fission. So remember, eukaryotic cells, if you can still remember your anatomy, they divide either by mitosis for the somatic cells or meiosis for the gametes. However, for prokaryotic cell, they divide by means of binary fission. It's so simple that, for example, this is the bacteria, then they will simply split, and then you'll now have two bacteria. And it can happen for every 30 minutes bacteria. Okay. All bacteria are prokaryotes. So that's a given fact. And archaebacteria are prokaryotes. So what are the archaebacteria? You can still remember them? The methanogens, the extremely halophilic, and the extremely thermophilic bacteria. So these are considered as the archaebacteria. Unlike eukaryotic cells, the cytoplasm of prokaryotic cells is not filled with internal membranes. In fact, you will not be able to find any nucleus in the prokaryotic cells. It's so simple, no? If this is a bacteria, you'll not find any nucleus, and instead of nucleus, what you can see here is a nucleoid. Okay, what you can see is a nucleoid. And what is a nucleoid? Nucleoid do not have nuclear membrane. You'll not be able to find any nuclear membrane. Okay, so the cytoplasm of prokaryotic cells is surrounded by a cell membrane. So just like the eukaryotic, they also have cell membrane, cell wall, and sometimes a capsule or a slime layer. So later on, I will introduce to you how a capsule or a slime layer would look like. But I just want to give emphasis now that the function of the capsule or slime layer is to prevent phagocytosis. So the capsule or the slime layer can prevent phagocytosis. So as what I've told you, it's so simple. So there's no nucleus. Instead, what you can see is a nucleoid. And genetic materials, chromosomes, okay, are merely suspended in the cytoplasm. Unlike in eukaryotic cell, the chromosomes are being enclosed inside the nuclear membrane. But since this particular cell doesn't have any nuclear membrane at all, so the chromosomes are merely suspended within the cytoplasm. Okay? So later on, we will be discussing what are the different parts of a prokaryotic cell. So again, let's discuss them one by one. Just like your eukaryotic cell, the prokaryotic cells would also have cell membrane that would have a similar structure and function with that of the eukaryotic cell membrane. Meaning to say, just like your eukaryotic cell, they are also semi-permeable. Okay, so they are also semi-permeable, and many enzymes are attached to the cell membrane where metabolic reactions take place. Okay, now chromosomes are also found in the prokaryotic cells. So we call it a prokaryotic chromosomes, which usually consists of a single long circular DNA and serves as the control center of the cell. Okay, unlike in eukaryotic cell, wherein the DNA is a double helix structure, it is a single long supercoiled circular DNA. So as you can see here, just a single supercoiled linear or circular DNA. Okay, so that's the structure of the DNA. So aside from the chromosomal DNA, there is also a plasmid, which are not part of the DNA. So that's the reason why we call plasmid as an extra chromosomal circular DNA. So it's an extra chromosomal circular DNA. So the cytoplasm of the prokaryotic cell is also a semi-liquid that consists of water, enzymes, waste products, nutrients, proteins, carbohydrates, and lipids. And just like your eukaryotic cells, this is where the action takes place because these components, are all required for metabolic functions. Okay. Unlike eukaryotic cells, you cannot find membranous organelles in prokaryotic cells because mostly what you can find here are ribosomes. Okay? However, the ribosomes of prokaryotic cells are much smaller. Okay, so take note that for prokaryotes, the ribosomes are known as is characterized as 16S. Okay? For eukaryotes, the ribosomes are described as 18S, wherein S stands for Svedberg unit, the unit of measurement in determining the weight of the ribosomes by means of ultracentrifugation. So prokaryotes, ribosomes is 16S, eukaryotes is 18S. So they are actually different. However, they serve the same function, which is for protein synthesis. So both of them would be important for protein synthesis. Now let's talk about the cell wall of the bacteria. So the cell wall of the bacteria is chemically complex, and this defines the shape of the bacteria. This is the reason why we can have cocci, we can have bacilli, or we can have spirillum because of the bacterial cell wall, which provides rigidity on the exterior of the bacterial cell. Now, the main component of the bacterial cell wall is known as peptidoglycan, sometimes known as murin, and it is only found in bacteria. Okay? So gram-positive, there are actually two types of bacteria according to gram stain. So if the gram stain becomes purple or blue or violet in color, then we call it gram-positive. If it's pinkish, then it is gram-negative bacteria. Okay? So we have gram-positive bacteria, then we have gram-negative bacteria. So gram-positive bacteria have thicker peptidoglycan as compared to gram-negative bacteria. Gram-negative bacteria they have a thinner peptidoglycan. Now, some bacteria do not have cell wall. Therefore, they do not have specific shape. Okay? Mycoplasma is an example of bacteria with no cell wall. Hence, they are pleomorphic. So when you say pleomorphic, they can assume a variety of shape. So if they are pleomorphic, it means that they can assume a variety of shape. So the structure of peptidoglycan is this. So peptidoglycan is made up of sugar plus protein component. Okay, so the protein component and the sugar, so alternating structure of N-acetylglucosamine and N-acetylmuramic acid. And they are being cross-linked by peptide chains. So that's the structure of peptidoglycan. Okay, so peptidoglycan is the main component of cell wall. Cell wall of bacteria. So we call it peptidoglycan. So as what I've told you, gram-positive bacteria has a much thicker cell wall as compared to gram-negative bacteria. Aside from that, gram-positive bacteria has lipoteichoic acid and teichoic acid, which is not present in gram-negative bacteria. Gram-negative bacteria, however, has LPS, lipopolysaccharide, and that is very important because this is where the synthesis of endotoxin is taking place. No? The lipopolysaccharide. Okay, so apologies in the delay. Ayan. So this is where the endotoxin is actually being synthesized and in gram-negative bacteria. Another difference, gram-negative bacteria has outer membrane. So outer membrane is not present in gram-positive bacteria. Okay? So let's talk about the different structure of the bacterial cell. Okay? So glycocolics. The term glyco means sugar and calyx means coat. So glycocolics means sugar coating. Okay? The term glycocolics means sugar coating. So there is another delay whenever I'm writing. It's not usually being shown right away. So my apologies for that. Okay, so trying to fix something here. Sorry, my presentation hanged and got frozen. So I'm trying to fix it. Okay, so my apologies for that. Um, I'm now using my personal data. Okay, so again, we call it glycocolics. So glyco means sugar, while calyx means coating. So it's called sugar coating. And of course, glycocolics, why it's sugar coat? It's because it is made up of polysaccharide structure. It's the reason why it's called sugar coating. And there are two possible kinds of glycocolics. Could either be called slime layer and capsule. So slime layer, if it is disorganized, we call it slime layer. If it's neatly organized, we call it capsule. So some bacteria possess glycocolics. So it's a slimy gelatinous materials produced by the cell membranes and secreted outside the cell wall. Okay? So slime layer is loosely connected to the cell wall, so it's not organized, while our capsule is highly organized. Okay? So example of organisms with glycocolics are Pseudomonas. So Pseudomonas has a slime layer. Okay? Whereas Klebsiella, Neisseria meningitidis, and Streptococcus pneumoniae, they have capsule. So capsule can prevent phagocytosis. What is phagocytosis? So when we say phagocytosis, it is the engulfment of WBCs such as neutrophil and monocyte. So neutrophil and monocyte can actually engulf neutrophil and monocyte can actually engulf bacteria, resulting to phagocytosis. Okay, so this is an example of Klebsiella pneumoniae. Okay, a negative staining technique. So when you say negative staining technique, the background is actually the background is dark, and the organisms are actually colorless, or the capsule is colorless. And how would you know if it's capsule? You'd be able to see a halo surrounding the bacteria. So this particular colorless structure here, this is actually the capsule. So is it a bad thing that bacteria has capsule? Yes, it's good for them but bad for us, because it means that our WBCs will not be able to fight them. So capsule therefore is considered to be as one of the virulence factors. Okay, it is considered to be as one of the virulence factors. Okay, now just like our eukaryotic cells, prokaryotic cells are also provided with flagella. So motile bacteria possess flagella, whip-like appendages composed of threads of proteins called flagellin. So flagella is important for motility. Is it advantageous for bacteria to have flagella? Yes, because they can move from one place to another, especially if nutrients are no longer available. So somehow we can see that flagella is advantageous for them, but again, it's bad for us. Therefore, it is also considered to be as a virulence factor. Now, do you know that we can also classify flagella, okay, according to their number and arrangement. So, for example, when we say Peritricus flagella, Peritricus flagella means that the flagella, okay, surround the entire cell. Okay? So when you say Lophotrichus flagella, there is a tuft of flagella at one end of the cell. Okay? So when you say Amphitricus flagella, one or more flagella at both ends. Okay, so that is considered to be as Amphitricus. Monotricus means that there's a single flagellum, By the way, flagellum is the singular form of flagellum. Flagella is the plural form. Flagellum is the singular form. Oh, yon. Again, so this is the Peritricus. And then this is the Amphitricus, Monotricus, and Lophotricus. Okay, so this is how they look like under the transmission electron microscope. So this one is an example of Peritricus flagella of a Salmonella. By the way, if you're familiar, Salmonella typhi can cause typhoid fever. Okay, so we also have the pili, another example of a hair-like structure observed in gram-negative bacteria. And the polymerized proteins or the protein components of pili is called pilin. So pili are thinner than flagella and they have a rigid structure. Unlike flagella, pili are not associated with motility. Okay, it's not associated with motility, but they enable bacteria to anchor themselves to surfaces. Let me give you an example. An example here is Neisseria Gonorrhoeae. Neisseria Gonorrhoeae is the causative agent of gonorrhea. So if you will be looking at their structure, they are diplococci and they have fimbriae or pili. And the purpose of the fimbriae and pili is to attach themselves in the epithelial cells. So for example, this is our urethra. This is our urethra. Okay, so because of the pili and the fimbriae, Neisseria gonorrhoeae can attach themselves at the urethral lining. So what will happen is that our WBC neutrophil will try to phagocytize them in the urethra. Therefore, there would be what? Pus. That's why in Tagalog it's called tulo. And the reason why they cannot easily be removed from the urethra even if you urinate, because of the fact that Neisseria gonorrhoeae have pili that will attach themselves at the epithelial lining of our organ. Okay, so again, pili is another example of virulence factor. So it is another example of virulence factor. Now, if the pili, for example, is, this is the pili, and it's responsible for the transfer of one DNA of the bacteria to another bacteria, then it is called sex pilus. Okay? And it's important for conjugation. So what do you mean by conjugation? So it is the transfer of DNA from one bacteria to another. So when the DNA is transferred from one bacteria to another, then it's called conjugation. So again, this is the TEM of a bacterial cell possessing both the pili and the flagella. So take note that pili are shorter as compared to flagella, which are longer. And they have different functions. So let's talk about the spores. Okay. Spores or endospores are not found in all bacteria. Okay. So only few genera are capable of forming atypical spores as a means for survival. Okay, so these are the Bacillus and Clostridium. So Bacillus is an example of aerobic spore former because they are capable of forming spore. Clostridium is an example of anaerobic spore former. Okay, and why they are important? because they are important for survival in extreme conditions, such as heat and drying. So if the conditions becomes unfavorable for bacteria, they will now start to form a spore. What it means, and this particular process is called sporulation. And sporulation is not reproduction, okay? So that's different. Okay, so what is sporulation? So for example, this is the cell of the bacteria. Okay, this cell is the vegetative cell. Vegetative cell. And this is the endospore. Under normal condition, you only have bacteria but no spore. However, if the condition becomes unfavorable for the bacteria, if the conditions become unfavorable for bacteria, then there is now spore formation. And this is what you call sporulation. Okay? And spores have shown to survive for many years and are resistant to heat, cold, drying, and most chemicals. So we cannot just easily destroy spores. Usually, one spore is produced in a bacterial cell, which generates into one vegetative bacterium. So, okay? So, this is the cell, okay, with one endospore. So, if the condition goes back to normal, the spores may be inactivated, and that's what you call germination. Okay? Spore formation is called sporulation. But when it goes back to its vegetative cell, we call it germination. Okay. What's the clinical implication of the spore? What's the clinical implication? The clinical implication is that some bacteria has spores such as Clostridium tetani. So if you are a nurse and and you will be dealing with surgical instrument, you cannot just boil surgical instrument because the endospore of the bacteria cannot be destroyed by merely boiling. Okay? And Clostridium tetani can cause tetanus. So how do we destroy bacterial endospores? So the only way that we can destroy them would be by means of autoclave. Okay, so an autoclave is steam under pressure. Steam under pressure, wherein you apply 121 degrees centigrade for 15 minutes at 15 psi or pounds per square inch. That's the only way for you to be able to destroy the spores. Okay, and how do they look like under the microscope? This is how they look like. See, this is how the spores look like under the microscope. It's another example of spores. Okay, so a terminal spore is located here, while a subterminal spore is located here. Okay, so let's differentiate. Eukaryotic cells contain true nucleus. Prokaryotic cells do not contain true nucleus. Eukaryotic cells are divided into plants and animal types. Animal cells do not have a cell wall. Plant cells have a simple cell wall. Eukaryotic cells contain membranous structures and many membrane-bound organelles. Prokaryotic cells possess no membranes other than the cell membrane that encloses the cytoplasm. Okay, so that's basic difference. Okay, so prokaryotic cell division. Prokaryotic cells divide by means of binary fission. That's what I've told you a while ago, wherein a simple cell splits into two. Before a prokaryotic cell divide, the chromosomes must be duplicated. And the time it takes for the binary fission to occur is called generation time. And generation time varies from one species of bacteria to another, depending on the growth condition. So for example, if the condition is optimal, Escherichia coli will have about a generation time of 20 minutes. What does it mean? So it means that every 20 minutes, Escherichia coli will double its population because that is its generation time in an optimal condition. Okay? So this is how simple binary fission is. So after the genetic material or the DNA is duplicated, there will just be a septum in between these two duplicated DNA, and they will simply split into two daughter cells. But because we are gender sensitive, we will no longer call it daughter cells, but we will be calling it as progeny. So there would be now two progeny. Okay, so let's talk about taxonomy. So taxonomy is the science of classifying living organisms. So taxonomy consists of classification, nomenclature, and identification. Nomenclature is very important, or scientific name is very important, because a scientific name is a universal name of a certain living organisms. So classification is the arrangement of organisms into taxonomic groups known as taxa. Yan. So in order for you not to forget the hierarchy, we have here a mnemonics. It's CDOFGS. King David came over for good spaghetti. So don't forget K for kingdom, D for division, C for class, O for order, F for family, G for genus, and S for species. Now take note that a scientific name is the genus and the species of a certain organism. So, for example, in Escherichia coli, the genus is Escherichia and the species would be coli. And we usually place an underline or you italicize it. So, the science of taxonomy was established based on the binomial system of nomenclature. So in the binomial system, each organism is given two names, like what I mentioned a while ago, the genus and the specific epithet. So taken together, both names constitute the species. So genus and specific epithet. So for example, Escherichia coli, Escherichia is the genus and the coli is the specific epithet. Therefore, the species is Escherichia coli, or that is the scientific name. So the genus is frequently abbreviated with just a single letter, E. coli. So E for Escherichia, provided that you have already mentioned Escherichia in your previous sentence. So for example, you have already mentioned Escherichia in your previous sentence. So in the succeeding sentences wherein you'll be using the same species, so instead of spelling Escherichia again and again, you simply abbreviate it as E. So E. coli. So the abbreviation ST is used to designate a single species. And if there are more than one species, we use the abbreviation SPP. Okay? So organisms are categorized into larger groups based on the similarities and differences. So in a 5 kingdoms scheme, bacteria and archaea bacteria belongs to kingdom Prokaryote. Algae and protozoan belongs to kingdom Protista. Fungi belongs to kingdom Fungi, or in some other texts, you'll be able to find Mycota. Kingdom plants belong to kingdom Plantae, and then animals belongs to kingdom Animalia. Viruses are considered to be as a non-biological entity because they are acellular. They do not have cell membrane. So other system of classifications do exist. So for example, the 5 kingdom classification was actually proposed by Whitaker. But Carl Woese Carl Woese uh, Carl Woese actually proposed the three domain system. So we have the archaebacteria, we have the bacteria, and we have the eukarya. All eukaryotic organisms belongs to the eukarya. True bacteria belongs to bacteria, while the archaebacteria belongs to the prokaryote. Archaebacteria would have a separate domain. So what's the basis of this domain system? So the basis is based on the differences in the structure of certain ribosomal RNA molecules among organisms in three domains. So they have to do the genotypic characterization by means of PCR. The most widely used techniques for gauging diversity or relatedness of organism is called rRNA sequencing. So ribosomes are composed of two subunits, diba? Like what I've told you, we have a small subunit and large subunit. So the small subunit is composed only of one RNA molecule, which is coded for a gene called 16S rRNA. So the 16S rRNA, I think I've already introduced that to you, belongs to the prokaryote, while the X18S rRNA gene belongs to the eukaryote. So that's how we determine how organisms are closely related to one another. So to determine how closely related one prokaryotic organism to one another, scientists compare the sequences of the nucleotide base pairs by means of PCR of the 16S rRNA gene from one of the organisms with a sequence of base pair of 16S rRNA gene from another organism. And then after sequencing the genes, we usually upload this in a software wherein we determine how related or how closely related one bacteria to another. Okay, so the more similar the sequence of the sequence of the base pairs, then it means that these organisms are more related organisms. So by the way, this guy is Charles Darwin, and Charles Darwin is considered to be as the father of evolution, and because of that, because of that, um, it is now clear. It is now much clearer how these organisms are related from one another. So, because of the three domains, we can say that eukarya is more related to our archaea as compared to the bacteria. Okay, so this is now the phylogenetic tree of life. So as you can see, as humans belong here, we are more related to fungi and plants as compared to bacteria because we belong to one domain, which is the eukarya. Okay, so there you have it, our discussion on the cellular structure and characteristics of both eukaryotic cells and prokaryotic cells.